Good morning, students. Today we will discuss about the phylum Coelenterata. In the previous lecture, we had discussed some of the silent features of the phylum Coelenterata, and the remaining one will be discussed in this one. Phylum Coelenterata is also known as Cnidaria, and the organisms classified in this phylum are termed as Cnidarians. The term Cnidaria is derived from one of the most characteristic cells of coelentrates, the stinging cells called cnidoblasts or Pneudocytes. Cnidoblasts are used for anchorage, defense, and for the capture of prey. And all these will be discussed in the upcoming slides with more details. Myself, Balram Sain, Assistant Professor in Zoology, Government Dunga College, Bikaner. Before starting today's lecture, we will just rebrush or revise our earlier lecture. Habitat. Coelentrates are generally aquatic and they are generally found in marine water, and although few specimens like Hydra are freshwater. If we talk about the habit, they are solitary or colonial. If we talk about the body symmetry, they shows radial symmetry. In that we have discussed that animal is cut across the diametric plane. The animal will be divided into two equal parts. The necessary condition is that it should be cut across the diametric planes, and there are many such diametric planes across which the animal can be cut. If we discuss about the level of body organization, for the first time in animal kingdom, cells associated to form the tissues in coelentrates. Hence, they exhibit tissue level of organization. The tissues are arranged variously to perform the basic functions in their body. Germ layers. Coelentrates are diploblastic in nature. That means two germ layers, ectoderm and endoderm, are present, which forms the epidermis and gastrodermis. With the help of the diagram, this is the epidermis and this is the gastrodermis. In between the epidermis and gastrodermis is the gelatinous mesoglea, which is not considered to be a true layer. Hence, they are termed as diploblastic. If we talk about the digestive tract in coelentrates, we can say it is of incomplete type. Incomplete type means there is a single opening which acts as a mouth and anus. This is the gastrovascular cavity, and this is the single opening which acts both as the function as both as mouth and anus. And this is the part with the help of which it attaches with the substratum, and this is the gastrovascular cavity, and it is opening along which the food particles are engulfed, and it enters into the gastrovascular cavity where digestion occurs, and after which the waste material are coming out through the same opening, which is acting both as the mouth and the anus part. Digestion. In coelentrates, digestion is of both types, that is extracellular as well as intracellular. Extracellular means in this type of digestion which is occurring in the cavity outside the cells is called extracellular digestion. This results in the breakdown of food into smaller fragments, and these smaller fragments of food are then engulfed by the body cells and digested within the food vacuoles, and this is called the intracellular digestion. Hence, the digestion of food is first extracellular and then intracellular in coelentrates. Skeleton. Some of the coelentrates, example corals, have a hard skeleton composed of calcium carbonate. Great Barrier Reef is the example of the skeletons of calcium carbonate. And if we discuss about the body forms, two basic body forms are found in cnidarians which differ in the structure and function from one another. These are the polyp and medusa. It can be explained with the help of a diagram. Polyp is a cylindrical form which remains attached to the substratum, whereas medusa is an umbrella shaped and it is a free swimming form. Some coelentrates exist only in polyp form, such as Hydra, Adamsia, while some have medusa form as the dominant form in their life cycle. Example, Aurelia, jellyfish. Whereas some coelentrates exhibit both forms, polyp as well as medusa. That example is Obelia. Those coelentrates which exhibit both the forms polyp and medusa forms show alternation of generation in their lives, and it is called as metagenesis. Alternation of generation. Alternation of generation means polyps alternate with medusa and vice versa, medusa to polyp and polyp to medusa. The polyps produce medusa asexually, whereas medusa forms the polyp sexually. This is also termed as metagenesis or alternation of generation. Polyps undergo budding, asexual reproduction to give rise to medusa, and medusa produce sperms and gametes, and over sexual reproduction to give rise to new polyps. Now we will discuss about the Pneudocytes or Pneudoblasts, which are the most characteristic feature of the phylum Coelenterata. These are used for anchorage, defense, and for capture of prey. With the help of diagram, we will explain about the Pneudocytes. Pneudocytes or Pneudoblasts or stinging cells. These are the oval shaped cells which are present on the tentacles and the body. These specialized cell bears a nucleus which is present near its base and is called as basal nucleus, and the stinging capsule, and this is the stinging capsule called as nematocyst. The capsule is filled with poisonous fluid called hypnotoxin, which is injected with the help of a thread-like tube structure. This thread tube acts like a piercing needle. The lower end of the thread tube is enlarged to form a butt which bears spines. These are the spines in some pneudoblasts, and these spines are meant to hurt the prey. Now, these are the sensory structures which sense any sort of danger or prey. Now we will discuss the mechanism of discharge of pneudoblasts. The sudden eversion of a thread tube of pneudoblast is caused by increased osmotic pressure. When the osmotic pressure increases in the nematocyst in this capsule, which is probably due to rapid intake of water. As a result, in increase of pressure, this slit is forced open and the thread tube turns inside out and it comes outside, and entire nematocyst explodes to outside. Under normal conditions, the discharge is brought by mechanical stimulation of the pneudocyte, such as contact with the prey. The thread tube, once discharged, cannot be withdrawn. It can be explained with the help of a video which I will be showing. Just wait.
Let's take a look at the stinging process in detail. A brush of your skin with a tentacle is enough to set the stinging mechanism of millions of cells in motion. The skin, as we see it here magnified 5,000 times, gives off a natural substance, which when coming in contact with a stinging cell, is collected by a sensor on the cell. The sensor then decides whether the contact is with another tentacle or a potential food source, our skin. The sensor passes the information up to the body of the cell, which triggers an enormous buildup of pressure within the cell. And when pressure reaches 200 atmospheres, the coil, as we saw earlier, now turned dark, is fired and penetrates the skin at 40,000 Gs. The toxic dart then spreads the poison throughout the skin, much like a multi-headed poisonous arrow. Now imagine this process happening with millions of stinging cells. The result? As you might guess, inflammation and a great deal of pain. So how can you protect yourself from these millions of stinging cells?
This was the video showing the mechanism of discharge of the stinging cells. Thanks a lot for paying attention, and in the upcoming videos we will discuss about the classification of